Okay, so thank you, Pat Walters, for joining us on the Data Science podcast of the Data Professor YouTube channel. So before we begin, could you provide us an introduction about what you do and who you are?
Sure. First of all, it's really great to be on the podcast. I'm a huge fan of the YouTube channel, so really happy to be here. So my name is Pat Walters. I am a computational chemist, occasional blogger. I work for a medium-sized, I guess, pharmaceutical company in Cambridge, Massachusetts. I've been working in various aspects of computer-aided drug design for the last 25 years. So, have been part of things since the early days of PCs up through the data science revolution. And I'm enjoying taking advantage of all the data that's out there and available now, and all of the exciting tools that are popping up through the open science data ecosystem.
Awesome. So what is your educational background? You mentioned about computational chemistry.
Yeah, so my undergraduate degree is in chemistry. After I finished my undergrad degree, I spent 5 years working as a chemist in industry. And during that time, I sort of moved out of working in the lab and running reactions and moving more into various computational aspects. After that, I spent 5 years working in industry. Then I went to grad school, got a PhD in chemistry, studied organic chemistry, wrote my dissertation on applications of artificial intelligence in identifying the conformations of small molecules.
Right. And so how did you actually get into AI? The thing is, what inspired you to learn that?
Yeah, I guess I didn't really even know what AI was, and this must have been sometime around 1985, 86. I walked into a bookstore and they had a book together with software on a programming language called Prolog, which was one of the first artificial intelligence languages. So just got that book, and Prolog programming was a lot of fun. So I started doing that, and then ended up that the company I was working for got acquired by another company, and they had a group that was building expert systems. So I learned to program in Lisp as part of that, got more involved in the AI aspect of things, wrote some code to do a variety of relatively simple machine learning using decision trees, techniques like ID3 and C4.5. And then as I moved out of grad school into the pharmaceutical industry, there were just more applications for machine learning. And then over the last 5 years, it seems like the whole field has really exploded.
Right. Actually, yeah. So as you mentioned, you're probably into machine learning, like even before the term data science was even coined in 2012. So back then, how was it like? I mean, are chemists a bit skeptical about AI, and are they really accepted back then versus right now?
I think it was harder at the time, maybe because there was less data, there weren't as many methods available. And I think data science hadn't really gotten as well integrated into the way that people work in industry and in academia. I think now, with the broad availability of a lot of open source tools, I think that there's a lot more interest in that. I think once people get their hands on these tools and they start to realize that it's not really all that complicated or all that mysterious, and once people get the opportunity to actually try this out with their own data, then they start to see where things work and where things don't.
Right. And you mentioned about learning to program, and recently I've seen that a lot of your blog are or into Python. Could you explain a bit about your experiences learning different languages?
Sure. Yeah. I mean, I started out in the early 1980s programming in BASIC, moved from BASIC into Pascal, moved from Pascal into C, moved from C into C++ and Perl. And then I guess I started programming in R maybe 15, 16 years ago, because that was really the first set of tools available for doing data science. I think R really started the entire concept of a data frame, which is now, when you program in Python and you program with a library like Pandas, data frames have become the accepted standard. I think, 15 years ago, they really weren't. So I started programming in R because of the availability of data frames, the availability to easily do plotting and to do a lot of data science and machine learning operations. But I think over the last 10 years, the Python ecosystem has really been built up to such a degree with things like scikit-learn, with pandas. I really haven't written any R code in the last 5 years, probably.
So currently, Python seems to be your go-to language so far?
Yeah, and it seems to be, it's kind of become the common language, especially in machine learning. So if you take a look at any libraries that are released in the open source world, I think, probably 95% of what you see is in Python.
Exactly. Actually, I came across several of your research publications, and you have several papers that goes into detail on AI, machine learning, deep learning, and how they are impacting drug discovery. Could you maybe explore about this topic, how AI is transforming, or is it just a hype for drug discovery?
Yeah, so maybe we can just start at the beginning of the drug discovery process. So early on, you want to be able to find a molecule that you can optimize. A lead molecule, a starting point for a drug discovery program. And I think there are a number of techniques now that enable you to do this. For a long time, we've used a technique called virtual screening, where we can screen through large libraries of available molecules. The thing is that the number of available molecules 5 years ago was somewhere in the neighborhood of 10 billion. I'm sorry, 10 million. And that was, on reasonable computing hardware, you could get through that in a day or so, typically. Now we've gotten to the point where there are more than 40 billion molecules that you can get readily. So it required us to use a different set of techniques. So one of the techniques that we've been using quite heavily is a method called active learning. So in active learning, what you do is, let's say that we're doing something like what we call docking, where you take a small molecule, you generate the 3D conformations of that small molecule, and then you look at how that molecule is going to fit into a protein binding site. So, that can take anywhere between a second and 10 seconds per molecule conformation to carry out that calculation. So it's not the sort of thing that you can do on billions of molecules. But what you can do is say, I've got a large collection of molecules, I will sample 1% of that. I'll use that to build a machine learning, or I'll sample the 1%. I will dock those into a protein binding site. Then what I can do is take that, use it to build a machine learning model to predict those docking scores. This machine learning model can process tens of thousands of molecules per second. So this way we can, by sampling a couple percent of this actual data set of 40 billion, we can then quickly model and get to a large number of hits quickly. So active learning has made it very easy for us to be much more efficient with the calculations. There's been a lot of work recently on methods for generating molecules. So it's a lot of ideas that are similar to things that have been in the popular press with tools to generate images. So you can say, great, I'm going to take a set of images, pictures of people, and then I'm going to use a neural network and generate photorealistic images that are not actual people. You can do the same sorts of things with molecules, where you can take a set of molecules, have a neural network generate new molecules, and even generate new molecules that meet specific criteria. It's an exciting technique, but I think that there are still a lot of caveats associated with that, and there's still a lot of work to do. Another area where machine learning is making a big impact is in being able to generate models to predict the physical properties of molecules, to predict their biological activity, to predict things like the pharmacokinetics of a molecule. So what's going to happen to a molecule when you then dose that into an animal? Is it going to be readily metabolized? So all of these areas are impacting drug discovery. That being said, there's still a lot of work to be done. I think we've only scratched the surface of what's possible at this point.
Right. So it appears that AI has become an integral part, one of like the pillar of computational drug discovery. Could we maybe explore a little bit about the major hurdles, challenges, or maybe the future trends that you see for AI in drug discovery?
Sure. Yeah. I mean, I think that there are still a lot of open questions. When I look at building a machine learning model, I kind of look at this in terms of three components. There's the data, there's the representation. How do you represent a molecule in a way that it can be processed by a machine learning model? And then there's the algorithms. So if we look at the data, the size of the data sets that we're dealing with in drug discovery is often hundreds to maybe thousands of molecules. Whereas if you look at people doing image analysis, they may have millions or even billions of images. So I think one of the challenges is being able to work effectively in this low data regime. Along the data side also, there is what we call the applicability domain. So if I build a model based on a set of molecules, how do I know, given a new molecule, how confident I am in the ability of that model to predict on my new molecules? I think there are challenges there. I think there are still a lot of challenges in terms of the ways in which we represent molecules. Right now, the vast majority of the work being done is just using what we call two-dimensional descriptors. So you'll have some representation that indicates the presence or absence of certain functionality in a molecule, or properties that are fit to experimental data. But one of the issues with machine learning is that machine learning is typically designed to come up with a relationship between an object and some observable. So if I've got a molecule and I've got whether it's going to dissolve in water, so I've got an object and an observable. The thing is, in chemistry, a molecule is flexible. So if you start thinking about dealing with molecules in 3 dimensions, then you've got to capture multiple representations of that molecule and associate it with a single endpoint. So this is a field called multiple instance learning, and multiple instance learning with molecules and in a chemistry domain, I think is still an area with a lot of open questions.
Right. So as you mentioned, multiple instance learning, is it that each conformation are given a set of vectors of molecular descriptors, and then they are used for machine learning? Is it like that?
Yeah. But then all of those vectors are going to be associated typically with a single experimental endpoint. So how do you develop, if you look at a typical machine learning algorithm, it's associating a single representation with a single endpoint. In multiple instance learning, what you're trying to do is associate multiple representations of an object with a single endpoint. So, there have been a few papers on multiple instance learning for developing quantitative structure activity relationships, but I think it's still a field with a lot of open questions.
Right. Actually, this is a very interesting area that I myself was pretty interested a while back because, like, for the most part, we noticed like that when we generate like 2D representation or even like a 3D representation, it would only capture one possibility of all of the possible conformation of the molecule. And so back then, I was thinking about maybe exploring the use of molecular dynamics in order to generate like trajectories of different conformation and possibly generate, like a lot of quantum mechanical descriptors out of that. But then never had the chance to do that. Is that multiple instance learning, as you mentioned?
You can. So you could also do multiple instance learning where you've got a set of frames from a molecular dynamics trajectory. So in molecular dynamics, you're observing the motion of the molecule. So you could take time slices of that and then capture some description from that set of time slices and then associate that. If you take a look at what most of the work that's been published on using molecular dynamics, what people are doing is just taking some aggregate descriptor. So you're really just taking an average over that set of frames. And from the work that I've seen, those aggregates don't really give you performance that much better than what you would get from a simple set of topological descriptors.
Okay. And would it also make sense to analyze the trajectory and maybe cluster the trajectory, because different trajectory might have like a different like shadow point or minimum, maximal in the conformational space? Is it much better to, like, maybe, for example, perform a PCA analysis of the conformation, and then based on the cluster, maybe for each cluster we sample maybe like the center portion of it? And
yeah, you can definitely do that if you're capturing this over a series. You should be able to get that clustering behavior out of that series. But yes, there are a variety of methods for clustering and creating a lower dimensional representation of a molecular dynamics trajectory. And then there are also trajectory techniques that are used to capture certain modes, slower modes that may not come out of something like principal component analysis. So there's a lot of exciting work that's going on right now from people like Frank Noe, who are now combining molecular dynamics and machine learning. So I think we're going to get to the point in the next 5 years where machine learning is going to be a critical component of a lot of physics-based methods.
Right, this is very exciting. And given the recent launch of the AlphaFold, which makes the accurate prediction of protein structures, and as you mentioned, I mean, if machine learning could also play a part in molecular dynamics, I mean, that would greatly enhance the capabilities of what it has to offer. What are your viewpoints of that?
Yeah, I agree. I'm incredibly excited by techniques like AlphaFold because I think that there is a large complementarity there with experiments. So one of the things that a lot of people are doing now is using AlphaFold as structures as a starting point to solve experimental structures using techniques like X-ray crystallography and cryo-electron microscopy. I think that as we move forward, it may be possible to capture more of this behavior and start using machine learning more toward capturing the dynamic behavior of molecules as well as the static behavior that you're seeing with techniques like AlphaFold. The other thing that I'm really excited about is that now there are multiple approaches, and it seems like each of these approaches has its own advantages. So there's AlphaFold, and then there's RosettaFold from David Baker's group, and there's the approach from Mohammed Al-Quraishi at Harvard. So I think that as these techniques become more widely used, there's going to be a lot of crosstalk, and this is going to expand their use. The other thing that I'm really excited about is all of this is out there as open source software, and as a real advocate for open science, I'm incredibly excited by the fact that we can all get our hands on these techniques, start using them, and start seeing where things need to be improved and where they can be used most effectively.
Right, exactly. Like prior to the podcast, I went through some of your publications and noticed that back in 2013, you published a paper promoting open science and the reproducibility, particularly for sharing code. Back at the time, I mean, there's not a lot of paper in QSAR or in Cheminformatics that would share their data and code. But then recently, I've seen a lot of more papers that are sharing their code publicly, which is a very good thing. But then again, there are some papers that are not yet sharing their code. So what are your thoughts about how we can drive open science to be more like a requirement for publication instead of, not sharing the code for that?
Yeah, I've been a big advocate for this. Actually, I wrote that paper twice. I wrote it, I think, once in 2013 and again in 2020. I wrote another editorial on this. And you're right, I've seen much more of a trend. And I think a lot of this has come out of people coming into cheminformatics and quantitative structure activity relationships from computer science, where it is expected that you release the code along with your paper. And I've found it frustrating because you'll go through a paper and often there are details missing and you can't reproduce what's there based on the description in the method section. The other thing is, it's just often difficult to tell, even if you are re-implementing something, whether you did it right, because you don't have all of the intermediate results. So I love the fact that we are now getting to the point where there is more and more of an emphasis on releasing code. So, this is what I do all the time. I'll get the code, I'll download it onto my machine, I'll try it out on one of my data sets, and sometimes I'll find things that don't work as well as they could, and then get back in touch with the authors. And often I find that people will then, I'll do a pull request on their code and they'll have made a change within a day or two. So I'm just really excited. I'm hoping that this gets to the point where not releasing code is kind of like smoking in public. It used to be socially acceptable. I'm hoping that as time goes on, it kind of just becomes the expectation.
Right. Like, for example, like in some of the publications of our collaborators, like, because we're mostly, like my research group is mostly computational biology. And so like some of our collaborators, when they send a manuscript for publication, most of the journals nowadays, they would require them to submit their original scanned image of like electrophoresis or mass spectrometry data along with the manuscript as a supplementary information. And so I definitely think in the near future, the same thing could, it is more easily implemented to just, upload it to a repository like GitHub or Zenodo or even to the publisher's own archive as well. So
yeah, and
very helpful for that.
Yeah, I agree. And I think there are also just positive things that we as a community can do to encourage that. So usually if I see a paper with code that I like, I'll just put out a tweet and say, look, this is a great method. The code is available. Give it a try. I try to occasionally, through my blog, highlight things in the open source world that I find interesting. The other thing that I really love is there's all kinds of great stuff coming out in just sort of the the general open source data science world, a lot of things that I see now on your YouTube channel. And and a lot of these are not necessarily chemistry related, but they're great tools that can enable us to make our lives easier.
Right, definitely. And so I was wondering whether the Lipinski Rule 5 in the pharma industry right now, in your opinion, do you think it's still an accurate indicator of drug likeness, or is there a better metric for measuring the drug likeness property?
Yeah, I think that it depends. I think in traditional small molecule drug discovery, typically, I don't think people adhere to the rule of 5 the way that they used to. But it's a reasonable heuristic, and it's the sort of thing that we look at, and you go, okay, yeah, that molecule, it's got a really high log P. We're probably going to have some solubility difficulties with that. But I think the other thing that's exciting is that we've got this whole new set of techniques that are moving us very far outside of the rule of 5. So if you think about things like targeted protein degradation, so there are these PROTAC molecules, which are big. They're often 1,000 molecular weight, which is way outside of the Lipinski rule of 5. And I think as a field, we've still got to now figure out what is it about these larger molecules that is going to impart reasonable physical properties and give us molecules that have good pharmacokinetics. So I think that a lot of those rules and, not even rules, but just guidelines and heuristics are still evolving.
Right, because this work was based back in 1997 by Christopher Lipinski. And in light of the recent advances in deep learning or machine learning, what are your thoughts about maybe revising the Rule of 5 and maybe implementing something using deep learning or machine learning in order to generate a more modern form of measuring drug likeness?
I think it's going to be tough, actually, because in order to build a good machine learning model, you need to have experimental data. And I think that there are just very few, there are examples, but there's a relatively small number of examples of molecules outside the rule of 5 that have become drugs. I think, as time goes on, we may be able to do this, but I think given publicly available data, I think it's going to be very difficult to do that. Perhaps if you're inside a large pharmaceutical company and you've got access to some of these data sets, then maybe you could start teasing these things out. But I think it's going to be several years away.
Right. So can we maybe zoom out a bit and explore the general topic of what is the tech stack in use in the pharma industry today? I mean, based on your role in the pharma company, in terms of programming language or software, either proprietary or open source.
Yeah, I mean, there's a variety of commercial packages that are being used for physics-based modeling. There are also a number of open source packages. One of the most popular for doing molecular dynamics is the OpenMM package that started in Vijay Pandey's group at Stanford and is now being the work is continuing through people like John Cadera at Memorial Sloan Kettering. I think, for the most part, in terms of machine learning, everybody, for the most part, is programming in Python. We do a lot with Jupyter notebooks because that gives us the ability to do a lot of exploratory work, to record the work that we've done, to integrate graphs and plots into that. And then it's nice that there are a number of open source packages now that are being widely used. So one of these is the ChemProp package that came out of MIT. There's the DeepChem package that came out of Stanford. So I think, for the most part, in terms of machine learning, it's an open source ecosystem.
That's great. Like for example, based on my own experience, like back in the days, we would be limited by several commercial vendors, like for example, by maybe companies like MOE or Schrodinger. That was back in the time when there weren't much open source software available. Like RDKit was not as powerful as it is today, or Jupyter Notebook was not in existence. And so, I mean, there has been a lot of advancement into open science, and so that makes, like accessibility by universities or academia or organizations that wouldn't have otherwise been able to afford such expensive software. Like for example, in our country, so like we do have funding, but then sometimes they're quite limited. And so like instead of spending a couple $1,000s on licenses, I mean, we could have better spent that on like hiring people or something more. I mean, if you could have like a trade-off between like just purchasing software or paying for publication so that it will be openly available. So, I mean, it's a very exciting time nowadays.
Yeah, I agree completely. I every week go through and, as I look through the literature, as I look through my Google Scholar alerts, or I look through new papers that are in ArXiv, I'm really excited by how much of this is being released as open source. I think one of the reasons that machine learning and drug discovery has advanced at the rate that it has is because of open source, and because people have been able to rapidly build on the work that other people have been doing.
Mm-hmm. Right. And another area is like open access publishing. Like back in the days, I mean, predominantly there will be papers published in traditional subscription-based form. And like when open access rolled out, like in Thailand, for example, they were a bit skeptical about papers paying to publish. So they're a bit wary about like for example, they had this the Bales list of predatory journals, and so like in the early days, I mean, they would be a bit skeptical about like open access publication. But nowadays, like even Nature or Springer or Wiley, they're transitioning many of their traditional journals to be a complete Go open access journal. And so, in our opinion, and also like there's a lot of publication as a preprint on ArXiv, Bioinformatics, or ChemArchives. And like, for example, in the data science community, I noticed that a lot of the papers that the aspiring data scientists are reading are actually archives, and they're not really coming from like Elsevier or Wiley or the traditional journal. And could you maybe explore about that a bit? Like in the pharma industry, is there any like support to go for the open access route, or is it really depending on the BI of the paper?
Yeah, I think it's going to really depend on that. It's going to depend on the field. I think biology papers are still going to end up in Science and Nature. But I am really excited about the fact that there are so many different open access journals. I'm actually part of the editorial board for a new journal called Artificial Intelligence in the Life Sciences, which is completely open source. It's a new journal, and actually for the first year, they're waiving all of the fees for publication. I love that, and I absolutely love ArXiv. I would say probably more than half of what I read these days are papers that are on ArXiv, and I'm really happy about the fact that Google Scholar seems to have figured out the sorts of things that I like to read. So now when I go into my alerts, I'll probably find that 60% of the papers that are recommended for me are things that are on ArXiv. And I found that a lot of people doing work on applications of machine learning in drug discovery, both in industry and in academia, are publishing a lot of papers on ArXiv. So
for ArXiv, do you think that it is an intermediate platform for sharing papers that are in the process of becoming published by the journal? Like, for example, it is going on peer review and is deposited in ArXiv, or are some paper deposited in the ArXiv and then abandoned, and I don't know. So in the pharma industry, which is the use of ArXiv?
Yeah, I think it's both. I mean, sometimes you just want to put something out there and you may not necessarily think of that as something that you're ultimately going to publish. But from what I've seen, it's a great way to get feedback before you submit your paper. So you put your paper out there on ArXiv. A lot of times I will either provide feedback directly on ArXiv or just contact the authors and say, you know what, here, I saw your paper on ArXiv, especially if they have code available. Then I downloaded your code. I tried this set of examples. Here's what I saw. This may be a little different than what you saw. You may want to try this. So I think it's a great way to just get feedback.
Right. Yeah, that's an awesome way of putting it. And actually, before I've also experimented with putting papers on preprint. They have it on PeerJ, but nowadays they don't really support that. And yeah, I really like the PeerJ journal. They have this unique way of you can buy a membership and then you could publish there for free. Like a lifetime membership. And it's unlike any other journal. And so it's a new way of publishing in open science. And so can we maybe move back to your endeavor as a blogger? So you have the Practical Cheminformatics blog. Could you provide us some history on how it got started? What inspired you?
Yeah, I started this about 3 years ago because friends of mine and people I'd worked with in the past or people I had talked with at conferences would just send me an email and say, I'm really interested in the Free Wilson method of doing structure activity relationship analysis, but I don't really have an implementation and, I'm not a great programmer, but I'd like to play around with this. I'd like to learn a little bit more. Can you point me in the right direction? And then I gave a couple of talks and people were asking me about this kind of stuff. So I just said, I'm going to try this. I'm going to try this once. So I just went out and, I got an account on Blogger. I put the blog post out and I got a really good reception from it. A lot of people reached out to me. It seemed like people liked what I was doing. So I put it out. I like teaching. I taught when I was in grad school. I've done a little bit here and there. I feel like that there is, as data science is becoming more of a factor in chemistry, I think that there's a lot of people who want to learn more and don't really know where to go. The other thing is the blog it gives me the opportunity to also vent on some things that bug me sometimes. So, I feel like people don't give AI and chemistry a fair shake. So I've written a little bit about that. I get frustrated sometimes because I review a lot of papers and people don't do a great job with their statistical analysis on the data. So I figured, okay, let me show you at least my view of the right way to do statistical analysis to compare methods. We see these papers all the time where people compare 10 different methods and then they just have this table there and they take and just put the one that's the best in bold. And then there's a whole bunch of other methods and you start looking and you're like, wait, all of these values look the same to me. So, how do you actually tell whether something is better or not. So I wrote a bunch of blog posts on various aspects of statistics. At the end of last year, I just had this whole collection of papers that I'd read that year that I thought were interesting. So I just wrote a blog post where I highlighted the interesting papers on AI and drug discovery in 2020. So, it's nice. It's just, I can just go out and write about whatever I feel like writing about.
Yeah, that's awesome. Actually, I think that you could also maybe make it as like a 10 simple rules for plus computational biology. So like maybe you could say 10 simple rules for what practical cheminformatics,
Yeah,
common
that's a good one.
mistakes in cheminformatics or something like that.
That's a good. Yeah, and a lot of times these are just things that I do on my own to try and learn more. So the last blog post I wrote was because the group at NCATS at the NIH had released a bunch of ADME data sets, and I just wanted to go in and play around with those data sets and take a look at them. And I figured, okay, if I did this, probably other people are interested in this too. So I'll just write it up as a blog post. So, half the time it's just some new method that I'm exploring, that I'm trying to learn about, or some data set that I'm trying to learn about. And I figure, might as well just put together a blog post and let other people learn about it at the same time.
Yeah, that's a great way of doing it. Yeah.
Yeah, it's been a lot of fun, and I've met a lot of people as a result of it, and it's given me a lot of opportunities. So.
Yeah, and also like I noticed that you have co-authored the O'Reilly book, Deep Learning for the Life Sciences, together with the group of Vijay Pandey. And could you share us your experience of writing the book? How did you prioritize your time, manage your time, and how do you make this work in light of your busy schedule working in pharma?
Yeah, I got involved in that because I had known Vijay Pandey for a long time. I had had a variety of people from his group as either interns or employees when I was working at Vertex Pharmaceuticals. So Vijay got in touch with me, said that they're writing this book. To be perfectly honest, my part of that book, I wrote a couple of chapters. So it wasn't something that took a huge amount of my time. The other thing that I was really excited about was, I learned most of what I knew about programming from O'Reilly books. If I move my camera here and show you, there's a whole bookshelf back there that's just filled with O'Reilly books. So, the opportunity to author an O'Reilly book was really exciting for me. But it wasn't a huge time commitment. I had multiple co-authors. A lot of them wrote a lot more than I did. And the other thing is that O'Reilly has their own Google Docs sort of system that makes it relatively easy to collaborate and enable the editors at O'Reilly to look through things. So it was a fantastic experience for me.
Right, awesome. And I think that a lot of people would be wondering, did you get the chance to select the animal on the book cover?
We didn't. Everybody asked me, what's with the chicken? And I have no idea what's with the chicken. O'Reilly chicken. So was not mine. Yeah, I guess the thing that I'm looking at now is there's a framework to build books with Jupyter. So you can essentially take something called Jupyter Books, where you can take Jupyter notebooks and put them together into a book that people can interact with. So one of the things I'd like to do if I ever get some time is start putting together a Jupyter book on cheminformatics that introduces some of the principles, but enables people to get their hands on things.
Yeah, that would be great. There's a lot of aspiring data scientists or students in biology or chemistry who are looking into learning machine learning. And I think like blog posts like from yourself or from other I'm not sure if I could pronounce it correctly. Iwaitobin.
Yes.
With the penguin. He has a lot of great
blog posts. Greg Landrum, who is the maintainer and author of the RD Kit, has a great RD Kit blog. Yeah, so there's a lot out there, but I'd like to get something that's together in a more systematic fashion where people could just start from the basics of how do we represent a molecule, and then how do you convert a molecule into a set of molecular descriptors, go through topics like clustering, similarity search, kind of explain, I guess to me, this should be a part of every undergraduate curriculum. I feel like, we've reached a point where every undergraduate studying physical or biological sciences should have an exposure to data science. I think it's become a critical element of everything that we're doing.
Yeah, exactly. And actually, like this year in my university, we experimented with teaching the very first Python course for the undergraduate student. And we also taught them about how to use pandas, how to use scikit-learn, for them to prepare data and also to build a simple random forest model. And so all of that was in 3 weeks' time. And so they went from like zero programming language to being able to build a basic model. And so I was very impressed by the performance of the student. But then there were a lot of uncertainty about the format, about the platform that we're using. And for the most part, I mean, there's a lot of concepts that they had to learn in such a short period of time. Like based on your experience working in pharma, like for example, if you have a new intern or a new scientist who's beginning to start to learn programming, how would you recommend them? How would they get started from your recommendation?
Yeah, I think that there are so many good tutorials out there now on the internet. There are all sorts of courses offered through places like Coursera. And then there are so many platforms out there now. So you can go, you don't have to have anything installed. You can just go on Google Colab or onto Binder and just pull up a Jupyter notebook and start experimenting. So I think what we need to do is just provide something that gives people a place to get started. So I like your idea. You start out, you give people a brief introduction to Python programming. You show them how to do things like, read a CSV file into a pandas data frame, and then how easy it is once you've got that to then do plotting and to do, to build a machine learning model. I did this online workshop for the Royal Society of Chemistry a couple of weeks ago, and I was so happy with the response that I got, by not only by the number of people who showed up, but by the number of people who followed up with me afterwards. And I had students contacting me and saying, "This is great. I really like this field. How do I get into this? What should I study?" So I think that there's a lot of appetite out there for people to learn this stuff, and I think it's going to be the key to science as we move forward.
Right. And so as we briefly touched upon the topic, like how would one start bioinformatics or cheminformatics, either coming from a biology background or coming from a computer science background? What are your recommendations?
Yeah, I think the first thing is to learn some Python programming. And there are plenty of resources out there for doing that. Then, learn to use Jupyter Notebooks, which is very, very simple and something you can get up and running with quickly. But I think it's just get out there and try things. So, I've probably got more than 30 blog posts out there, and there's plenty of code there and things to try. So as I'm putting things together, I'm trying to kind of come up with more of a learning path for people who want to get into cheminformatics.
Right, perfect. Can we also explore the question of like, what is the day in the life looks like for you?
My day in the life is going to a lot of meetings because I have a relatively large group. But I think, the day in the life for the typical data science, computational chemist in drug discovery, a lot of that is working in teams. So you're working in a team with, you're a computational chemist, you're working in a team with medicinal chemists, with biologists, with structural biologists, with cell biologists, and you're trying to design the next set of experiments. The way that I look at what we do in computation is we're doing an analysis that enables us to design the next experiment. And then once we have that, we're going to collect that data and use that to design subsequent experiments. You know, when people ask me, what do you need to succeed, I think in computation, in drug discovery, it's kind of this intersection of a Venn diagram. The first is having the domain expertise and having an understanding of chemistry, biology, biochemistry. I think you need to have the other component of that Venn diagram is the hacking skills and knowing how to program and being able to access data. And then I think the third piece of that is having the knowledge of math and statistics. Because I think without a solid grounding in statistics, you can fool yourself and you can think that your machine learning model is telling you something that it's not. So I think it's really having a firm grounding in all three of those areas.
Right. And another question that I like to ask during the podcast is, what is your favorite machine learning algorithm?
Oh, that's a good question. Lately, and it really depends, and it depends on the task, and it depends on what I'm trying to do. I think if you ask me for my favorite, and my favorite last week was different than it is this week, it'll probably be different next week. But lately, I've been dealing with some very large data sets, so I really like GBM, light gradient boosting machines, because it's super, super fast and really easy to install. I really like XGBoost, but I spend a lot of time working on a Mac, and installing XGBoost on a Mac can be painful. So I like GBM because it's fast. It seems to give me results that are relatively equivalent to what I would see with a method like XGBoost or Random Forest. And it's relatively easy to get things like descriptor importance out of. But then again, there are other cases where I might like, a graph convolutional neural network. So it really depends. And I think it's the way that I look at the work that we do, it's sort of being able to pull the right arrow out of the quiver. And I don't think that there's any one algorithm that's perfect for every situation.
Right.
Now, if you have to ask me about my favorite Python library, it's Pandas. It's like every week I learn some other cool trick that I can do with Pandas. And this is one of the things that I like about blogs and YouTube channels like yours, because I look at those and I always, I'll be on Medium or something and I'll see, I always look at the one that's like 10 cool tricks with Pandas. And there's always the one that I didn't know. So
right. Or 10 things that you didn't know that Pandas could do or something like that, right?
Yeah. No, it's just Wes McKinney should get the Nobel Prize for writing Pandas, because it's
right.
It's completely transformed the way that I work.
Right. And have you ever tried Streamlit library?
Yeah. I don't do that much where I build GUIs. So I've read a lot about Streamlit. I've done a little bit of programming with it, but I haven't really had the chance to dive in.
Right, because they have a lot of cool cheminformatics related components. Like it's like a plugin. Like for example, you could have like a multi-grid where you could display a grid of molecular structures, and they have like a plugin where you could display a three-dimensional structure, like from JSMO. Yeah, so you could display the three-dimensional of the protein structure. And yeah, there's a lot of cool things done by another pharma company. I think it's called Rivery. And
Reverie Labs. I know those guys very well.
Right. So they've written like maybe 2 to 3 blog posts showing how to use Streamlit to make cheminformatics app.
I'm making a note right now, so I know what I'll be spending my weekend doing.
Okay, cool. And Yeah, so can we explore the topic of the interface between education and cheminformatics? Like in light of the current pandemic and the online education, or even like the absence of in-class education, in your delivery of cheminformatics education via blog posts or maybe by teaching on Zoom, what are some of the challenges that you think we're facing and how can we tackle that?
Yeah, I think it's hard because you do lose a certain interactive element. So a couple weeks ago when I was doing this seminar for the Royal Society of Chemistry, there were just this stream of questions coming through the chat. I think I could have spent the entire time just answering those questions. But that being said, I think so I think one of the biggest problems, especially with online learning with cheminformatics, you've got people who are coming with a very limited background in programming. So you give them a Jupyter notebook and they start typing away, and then they try to change something and it doesn't work and they're just getting some exception. And if they don't know what to do, and then they have to then put some post into a forum and they have to wait a day to get a response from me, that I think is going to slow things down, as opposed to being able to just sit there in a classroom and say, oh no, you just need to put another square bracket over there. That being said, I'm still, I'm happy with the fact that there are resources out there that people can try things. I'm really happy with the fact that there are a lot like I tend to follow the ArXiv mailing list, and it's a tremendous community. I'm constantly impressed with the fact that people will post a question and then it seems like the rest of us are in a race to see who can answer the question first. And usually people will have an answer back sometimes within 5 minutes, and I've done the same thing. I've had a question, I'm not sure about this aspect, how do I do this? And I'm always just blown away by how quickly people post things. I'm really happy with the feedback that I get sometimes from my blog posts. People will say, oh, you posted about this way to do this, and I think that this other way is better. So yeah, I'd like to get to the point where we had something. It'd be great if there was just some, cheminformatics Discord server that you could go to and you could just post questions and give the community more of an ability to respond.
Right, or something similar to the Stack Overflow.
Yeah.
Right. Okay, so I think we've covered a lot of topics for this podcast, and I'm very thankful that you have accepted to be on the podcast today. So before we end the podcast, could you share with us like how could the audience reach you or contact you or maybe read your blogging platforms on which media? So
Yeah,
how
sure.
can we contact you?
Yeah, I have a blog called Practical Cheminformatics. So that's practicalcheminformatics.blogger.com. I'm on Twitter. It's at WP Walters. So usually the best way to contact me if you have any questions is just DM me on Twitter.
Okay. Yeah. So thank you, Pat Walters, again for being on the podcast. And so probably maybe in the future, we could have you back on the show.
Yeah, no, thank you. This is great. I really appreciate the opportunity. And like I said, I am a huge fan of the YouTube channel. So really happy to be a part of it.
Yeah. And I could say the same to you as well. So I've been a long-time follower of your blog posts, and it's been very helpful for some of the videos that I've also created. So actually, there was one video where I cited in the Jupyter Notebook, one of your blog posts about using the Delani dataset for solubility prediction, and how you reproduce the result from the original research article. And that was a very great article that you have written.
Thanks. Yeah, I'm getting motivated now to try to do a couple of YouTube videos to cover some of the things and go a little bit beyond what I've done in the blog post. So we'll see how that works out.
Right, that would be great. And maybe we could collaborate on that as well in the future.
I'd love to.
Okay. Yeah. Okay. Thank you and bye-bye.
Bye.